Going to be by John
Edward.
Edward Green.
I have too many initials, so I apologise, it's confusing.
And he's from the University of Adelaide, and he's going to speak about mathematical models for cell-extracellular matrix interactions in tissue development.
Okay, thank you very much. I'd like to begin by thanking very much the organisers for giving me the opportunity to talk to you this afternoon. It's been really nice to be at this workshop and see some of the really interesting experimental work that people have done and hear about kind of the modelers who've been getting really into the details of the biology and producing some really kind of very nice simulations of what's happening in biological systems. I'm afraid I don't have any of that sort of thing in my talk, so unfortunately I have to start by saying please lower your expectations. So just to begin with, I think traditionally, because I'm a mathematician, I'm going to put all the acknowledgements at the start. So the work that I'm going to be talking about today is a collaboration involving Rosemary Dyson, mainly from the University of Birmingham, also Helen Byrne and John Whiteley from Oxford, and Anna Soto and Carlos Sonnenschein from Tufts University, who are biologists who really kind of got us started on this piece of work, although it sort of subsequently went off, I think, in a slightly different direction to what they might have expected and perhaps even hoped for. And of course, finally, I must thank the Australian Research Council for putting up the money. So those of you who are paying attention might have seen that while we were eating our lunch on Monday, we had a quick change of Australian Prime Minister, which seems to now be a sort of almost annual tradition. Anyway, so my talk is really, I guess, focusing on extracellular matrix and how cells interact with it. So sort of the idiot level at which I operate in terms of biology is that the extracellular matrix, or the ECM, is a kind of a protein network in which cells live when they're in vivo. And it can contain lots of different kinds of components depending on what kind of tissue that you might be interested in. So things that are quite common would be collagen, fibrin. If you're looking at cartilage, things like proteoglycans are important, and so on and so forth. So it can be very varied. Most of the problems that I have been interested in, and including this particular work, involve trying to grow cells in vitro. And so because collagen is such an important component of the extracellular matrix in vivo, well, in order to try and mimic a more kind of realistic and natural environment for the cells, often people are going to use collagen gels to seed their cells in. And so what we've got at the top there is a picture of what the structure of a collagen gel is like. So the square is sort of 70 microns along each side. And I think it's fair to say that a sort of representative cell size would be about 10 microns, so you can see that if you dump a cell down in there, it's going to be in contact with lots and lots and lots of different fibres. So the next slide is where I have a bit of trouble because the movie was not working, but I can come out of here and play it for you separately. So what we've got here are essentially 3 clumps of cells top, and then at the bottom on the 2 sides, 3 clumps of cells in a collagen gel, and they're, I think they're a kind of cancer cell if I remember correctly, and what they're doing is they're moving and they're proliferating, and as they're doing this you can probably see they're pulling on the collagen, and you start to get these kind of tracks forming in the fibres in the collagen. You get this kind of alignment effect, and then you can see that the cells seem to be kind of preferentially moving along those tracks. So this is a movie from a paper by Sawney and Howard, so I think this is probably about 12 years old, something like that. But just kind of indicates, I guess, the thing that I'm interested in, which is this interaction between the mechanics of the gel, so you have this kind of fibrous alignment effect, and how this can perhaps affect what the cells are doing. Okay, so let's stop that now. So yes, so essentially what I was kind of wanting to say about that video and sort of have you take away from there is that really, yeah, the cells are interacting with the extracellular matrix structure, and they're changing it because they're exerting forces. They can also, the cells can do things like they can secrete and degrade components of the ECM. So there's this kind of dynamic interaction, excuse me, between the two. Okay. No? Okay. That's not going to work either. All right. So this is this next slide is kind of just to give you a bit of an understanding of where we started on this work, and it actually started about 6 years ago when the Virtual Physiological Human Project, which is a EU-funded project, had a study group in Nottingham. And so one of the problems was brought along by Anna Soto and Carlos Sonnenschein from Tufts, and this is one of their pictures, basically what they were interested in trying to explain. So what they were doing was they were taking a particular cell type, which I think was a mammary epithelial cell, and they were seeding their cells in a gel which was a combination of collagen gel, but also includes matrigel and culture medium. So what they found was by varying the composition of their gel, they were getting a variation in kind of the cell morphology that you could see when the cells were grown in these gels of different compositions. So decreasing the matrigel concentration, which is kind of, I guess, making the collagen a sort of more predominant component, you went from having these acini, which are kind of little balls of cells, to having these kind of more elongated structures, which they called ducts. And so the study group problem was really trying to understand why this was happening and develop some kind of model for it, perhaps. And so with sort of 5 days on the study group, we decided that we would try and explore the hypothesis that this was connected really with the change in the mechanics of the gel that they were using. This kind of change to a more fibrous kind of mechanics was perhaps driving this change in the structure to from acini to ducts. So obviously one can argue about whether that hypothesis is true or not in this particular case, and I wouldn't like to say either way, but we just wanted to explore this as a possibility. So the ECM generally, we know in a variety of different situations, plays an important role in regulating cell behaviour, so the way that cells move, whether they proliferate, how much they proliferate. And so the feedback between the cells and the ECM is believed to be important in tissue development, in processes like wound healing, invasion of cancerous cells, all these kind of things. So essentially what we undertook in this bit of work, starting with the study group, was to look at developing a model to explore the role of cell and extracellular matrix interactions in patterning tissues. And we wanted to include the kind of complicated fibrous mechanics of the extracellular matrix in some way and look at how that might feed back on cell behaviour. So here was our basic kind of idea, that you have, well, your cells are sitting in some kind of fibrous material, and, well, as the cells are exerting forces on the material in which they're sitting, that's changing the fibre alignment. In turn, the fibres are helping to guide cell movement. Of course, as they move along, then the extracellular matrix is deformed, and then, of course, well, that changes the fibre alignment and thus feeds back into the movement. And so we were wondering, well, is this kind of feedback mechanism sufficient to be able to give you some kind of patterning? And if so, what sort of patterns can you get? And so we were trying to come up with a mathematical model that we could use to kind of explore this idea. So I hasten to add that this is all kind of very speculative. There's not really a very deep connection to any real biology here. Okay, so here is what we came up with for our model. So we decided to look at using a multi-phase kind of model, and so everywhere in what comes up in the following, we're going to have things that are to do with cells, denoted by an N or an N subscript. Collagen is going to have a C subscript, and then the other stuff in the gel that they were using, which is some combination of matrigel and culture medium, which I like to think of as water because that's a simple thing that I can think of. We're going to call that just generally medium, and that's going to have a subscript M. And for simplicity, we're going to assume that all of these things have an equal density. So then we've got volume fractions for each thing. So the volume fractions are denoted by phi with the appropriate subscript. And assuming that we've got no voids, so there's no kind of holes in this thing, all of those fractions obviously have to sum to 1. And then similarly, to keep things simple, although it may not necessarily be biologically realistic, we're not going to consider cell proliferation or death or extracellular matrix production or degradation. So these things are just kind of being rearranged under our hypothesis. They're not being produced or decaying. And so then for each phase you have a mass conservation equation of this form. So for each of the cells, collagen and medium, they're going to have this kind of conservation equation where the V's, Vn, Vm and Vc are the relevant velocities of each thing. So far, so very simple, I think. We're just conserving mass. Well, the real sort of, I guess, a difference in this model compared to a lot of the multi-phase models that are out there for things like cancerous tumours and other similar kind of situations is that we wanted to take into account somehow this fibrous nature of the collagen gel. And we wanted to do this in a kind of continuum sort of way because of just the sheer kind of number of fibres involved. It didn't seem practical to be able to track each individual fibre when you're looking on a sort of scale of hundreds of cells. So we borrowed an idea which will be familiar to people who've looked at things like liquid crystals, which is the idea that we have what's often called a director field. So the director is just a unit vector, essentially, that at each point in our collagen gel is going to tell us the direction of the fibre alignment at that point. So one kind of very big assumption there is that at each point I can tell you one definite fibre alignment, okay? It might actually be more realistic to say that at each point, well, there's a distribution of fibre alignments and I have some probability of finding a fibre, you know, at some angle alpha. But in order to kind of keep things a little bit simpler, well, we just assumed that there would be a single alignment at each point. And then we assumed that as the collagen is moving around, well, the fibres are just advected with the velocity of the collagen. And so when we kind of plug that assumption in, what comes out is the equation that we see at the bottom there. So our A, this is a unit vector, so it always has length 1, and it has the following evolution equation. So that's just essentially saying that our fibres are reoriented by a combination of stretching and kind of rotating with the local velocity. So then for each of our materials, we've got three things in our model, essentially cells, collagen and medium. We need to sum up their kind of mechanical properties that's encapsulated in the stress tensor for that particular material, so that tells us how it responds to forces. So that's going to be summed up by this stress tensor. Each phase, as it's moving around, will experience the drag from the other phases if they're not moving at the same velocity. And then finally, we're going to have cells exerting some kind of forces on the collagen. But again, for simplicity, we're going to assume that they don't exert any forces on the medium. Possibly, again, not realistic, but but for simplicity, and this model does get actually quite complicated, we decided just to focus on the cell-collagen interaction. So this is the momentum balance for the cells, and it's kind of typical of what you see from the other phases as well. Excuse me for a moment. So the stress in the cells is first term, and then we have cell collagen drag, cell medium drag. We've got force from the collagen, so cells exert forces on collagen, so collagen exerts the equal and opposite force on the cells. And then because we're using this kind of mixture theory mathematical framework where you have volume averaging, from the volume averaging you get this final interfacial term, which I don't really want to go into too much. The one thing that I would point out here is that because the collagen has this preferred direction, which is to do with the fibers, the drag also will be direction dependent. And so what we've got there for the KCN, that is actually a tensor rather than just a constant, because it has to take into account whether your drag is acting in the same direction as the fibers or not. So that's what happens for the cells, and then very similarly we've got the expression for the collagen, and then finally for the medium, again very similar, that we have these two drag contributions, and in the case of the medium we don't have a cell force contribution because we've just assumed that that isn't present for simplicity. And like I said, with the drag we do have to take into account that because the collagen is not isotropic, the direction the two things are moving in does matter, and so you've got an anisotropic drag between the collagen and the other components. So then, well, essentially our model is just what I've shown you so far. It's a combination of mass conservation and momentum conservation, but then of course you have to actually specify what the functions are for each of these materials. So what is the stress-strain relationship for cells, collagen, medium, and so forth. And so this is, to make a start with the cells, we're treating everything pretty much as a viscous fluid. So the cells are going to be a viscous fluid. So we have, in the top equation there, we've got P, which is the pressure. We've got this slightly odd gamma term, gamma star term, which I'll come back to in a minute, which is not kind of conventionally there in a fluid. And then we've got the 2 mu n term, so mu n is just the viscosity, and then we have a kind of bulk viscosity term at the end. So the gamma term is essentially because we're saying that cells are a little bit different to a standard viscous fluid in that they can want to clump together. So this is how we would imagine if there wasn't any kind of fibrous material that you would get these kind of clumps in the acini. And so what we're saying here is that the cells have some preferred volume fraction, which is this capital phi here, which they will try to attain. And so if you have them too sparsely packed together, they will try and aggregate, and if you have them too densely packed together, they will try and push apart. So that's what that gamma term there is doing. And so this is actually taken from a model by Chris Brouard and Helen Byrne, which was originally for growth of tumour spheroids. So that kind of assumption there was that the cells wanted to kind of clump together unless they got either too packed or too sparse. So the medium, we're just going to say that's a simple viscous fluid. So we just have the kind of very standard fluid equation there, no extra terms at all. And then finally we have the law for the collagen gel, which we've because we have fibers in this material, well, the fibers are going to affect the mechanics of the gel, right? If I try and pull the gel along the fiber direction, I expect its properties to be different to if I try and pull sort of transverse to that. And so we used a quite simple model for an anisotropic material, which is this transversely isotropic viscous fluid model. So transversely isotropic just means that there's one kind of preferred direction at each point in our material, which is the fibre direction, and every direction right angles to that, everything is kind of the same. So how did we get this kind of expression? Well, the first three terms, hopefully people will recognise, are just the standard terms that one would have for a viscous fluid. So the kind of all the nastiness is in these extra terms, the mu1 star, mu2 star, mu3 star and mu4 star. So how did we get these things? Well, what we did was to restrict ourselves to a relationship which was linear in the rate of strain. So that's linear in this EC, which is the rate of strain tensor. And every constitutive law has to obey certain invariances, so it has to be invariant under translations and rigid body rotations. And if you impose those conditions, then this is what you end up with as the simplest thing that obeys those invariances and is linear in E. So that was determined, I think, back in the 1960s by Ericsson, who was interested in these kind of liquid crystal sort of problems, and similar kind of things have also been used by Lee and Ockenden for looking at the mechanics of fibre tufts in the textile industry. So this is something that's kind of had many different guises, I guess, over the years. So the meaning of those those four parameters, just briefly. So mu1 kind of represents a tension in the fibre direction. mu2 is related to the extensional viscosity in the fibre direction. So if I remember correctly, mu2 plus 4 times mu3 is the actual extensional viscosity. Mu3 itself is the difference in the shear viscosity between the fibre direction and transverse to the fibre direction. So it's kind of the extra shear viscosity when you're shearing in the direction along the fibres. And mu4 is kind of an additional sort of anisotropic contribution. I guess it's similar to a bulk viscosity because it involves this divergence of E. Okay, so there's our constitutive laws for each of the three components, and then similarly we have to cook up some kind of sensible form for what our drags are going to do. And so, well, because the cells and medium are isotropic, well, the drag coefficient there is just, we can just have a constant drag coefficient, and we're going to assume that the drag is actually some constant times the product of the two volume fractions, so that if there's one of those constituents is not present, there's obviously no drag, and this is just a simple way of kind of achieving that. So that's what we set there. For things that involve collagen, of course, like I said, we've got a slightly greater difficulty because we're going to have a different degree of drag along the fibre direction to what happens transverse to that. And so we're going to assume that actually it's easier for cells to move along the fibre direction, and we're going to say that if you do that, well, the effective drag coefficient has this similar form with this capital D, similar to what we just saw. And if you were trying to go normal to the fibre direction, you would experience a greater resistance. So then, in order to get the right kind of behaviour, we set up our drag coefficients like this. So when you can see that when you're moving along the fibre direction here, this term would be 1, and so you'd subtract off that extra contribution. Okay, so that's a lot of kind of setting up of equations, and I've still got one to go, unfortunately. So now we need to finally determine what is the force going to be between the cells and the collagen. And so the idea that we had was that if I'm standing at some point x in the collagen, well, the force that the collagen at that point x experiences is going to be kind of the sum of all the pulls from the surrounding cells within some kind of sphere of influence. So each cell has some distance over which it can experience the over which it can affect the collagen, and so then at each point in the collagen, well, the force I experience is just the sum of the pulls from the surrounding cells. We're going to assume that the force depends on the direction of the fibres. So if I'm kind of at a point where there's a cell sort of just over there directly on a fibre, then I'm going to say that that exerts a very strong pull, whereas if there's a cell over here which is not on the same fibre as me, then that exerts a lesser pull. So kind of force transmission through the fibres is more effective, is the idea. And so what we do is, well, we decided we're going to look at having some force that is kind of an integral like this. So this looks very ugly, but what's going on here is basically this integral is just summing up all of the pulls of the surrounding cells. That's what it's doing. It's effectively a sum. So we have two extra terms here which I need to explain. So first of all, there's this F term, which depends on the distance between the point I'm sitting in the collagen at and where the cell is. And so this is just a way of having my force fall off with distance, so that cells further away have less effect than cells near me. And then, well, this G term is to do with the fibres. So what I'm saying is, well, if I look at this vector x minus x prime over the modulus of that vector, that's the unit vector that is pointing in the direction connecting my collagen point to where the cell is. And I take the dot product of that with A. So if they're aligned, that gives me plus or minus 1. And if they're at right angles to each other, it gives me 0. So this is a way of trying to take into account the sort of directionality of the pull in a crude kind of way. So the one thing that I've got to have in terms of this G is that, well, I want to have G be invariant under A going to minus A. So the idea here is that, you know, if a fibre is pointing vertically, it doesn't sort of matter whether it's kind of up or down, right? Both of those directions look the same to the cell. So plus A has to be the same as minus A in this function. And so what we did was we simply squared it, because that was a simple way of achieving that. Okay, so that was a pretty horrible kind of force function, and so we decided we would try and make things a little bit easier by assuming that in fact this sphere of influence of the cells is just pretty small, and so we can then expand our horrible integral as a Taylor series and integrate term by term. And after we've done that and sort of slogged through some rather ugly algebra, then we end up with a force function that looks like this. So it depends on things like derivatives of the cell and collagen density and similarly directional derivatives of those things. Okay, so finally I think we've sort of got the model in place at long last. And then, of course, well, you've got a horrible system of nonlinear equations which are quite complex, and certainly I can't look at them and write down a solution easily. So we decided to do some kind of quite radical simplifications to see if we actually could get anywhere analytically. So the first thing we did was to ignore the anisotropic drag components. We threw that out, so we said that the drag is the same between all of the components, no matter what the direction is. And then we said, well, let's look at the idea that you have only a few cells seeded in your gel, so the volume fraction phi n of cells is small, and then can we do anything by kind of using that assumption, expanding everything out in this kind of way? Does that kind of give us any insight into what's going on? And again, one ends up with some really horrible algebra to do, but we end up with an equation that looks like this. And so this is really kind of an advection diffusion type equation for our cells. So we've got a diffusion term is this first term over here on the right of the equal sign. It's an anisotropic diffusion, so D there is a tensor, and we've also got these effects which depend on gradients of the collagen. So actually this term here is kind of like a haptotactic term, which is actually driving you down gradients of collagen. So in haptotaxis models, I think I've seen probably more models where you are driven up a gradient of collagen, but under the assumptions that we made about our forces, in this case, what comes out is that you get driven down. So it is not entirely unlike some of the other kind of models out there for these kind of problems, sort of like the mechanochemical Murray type models. And here is just our diffusion tensor D, so you can see that this depends on the direction. Okay, so having kind of at that point thrown up our hands in horror at being able to do anything analytically, we went and enlisted the help of John Whiteley, who's a numerical analyst in Oxford, and asked him if he would be able to do some simulations for us. So to keep things simple without kind of throwing out the whole interesting part of the anisotropy, we were looking at just a 2D domain with periodic boundary conditions. And so what we did was essentially we made some guesses about our parameter values, and we got a set which we decided to call the control parameter values, which would give us some nice aggregates. So we've turned off here pretty much all of the anisotropy kind of parameters. So you can see mu1, mu2, mu3, mu4. These are actually all dimensionless now, but they are the equivalents of the anisotropic mechanical parameters that we had before. So we turned all those things off. This lambda here is always non-dimensionalized to 1. This is about the essentially gives you a measure of the force between the collagen and the cells, and so the gamma was that parameter essentially in the cell relationship, which is kind of the cell's affinity for this close packing density. So by adjusting this gamma, you can make them want to pack together more or less. So with our control parameters, we have that kind of ramped up quite high, and so we started with a situation like this, where all of our fibers are aligned parallel to the x-axis, and we have some little perturbation of our cells, so that sort of bottom left and top right there's slightly more cells, and then we sort of run the simulation and see what happens, and eventually it settles down to this kind of situation where you can see on the top right there you've got, well, our two patches of cells that have formed nice kind of circular clumps. There's a little bit of kind of realignment of the fibers around those clumps. They've sort of been kind of pushed outwards a little bit, I guess. But yeah, so this was kind of like our sort of our base behavior, and then we thought, well, what we'll do is we'll kind of look at the effect of changing some of these things and just see what happens, see what kind of patterns that you can get. So it's I'm afraid of a kind of a bit of an unscientific process. It's really just playing because we didn't know what things this model would do. Question?
Yes. Do I understand correctly that if you turn off all the muse, then it's only the passively advection and rotation of the fibers that you see here?
So if you turn off the muse, essentially everything just looks like a viscous fluid. So you've got cells in some kind of viscous soup.
Yes, and then the A vector reflects only the pulling direction of the cells.
Yeah.
That's it, and there is no feedback on the cells.
Yeah.
No change of their environment when they turn A.
Yeah.
Okay.
So so that was the control kind of situation. So then we said, well, what happens if you start with, instead of your fibers oriented parallel to the x-axis, what if you put them at 45 degrees? And so, well, I think sort of predictably, or what we expected anyway, you see that the two clumps start to kind of elongate along this fibre direction. And so now I can't show you this movie, unfortunately, because it doesn't work on this computer, but I can show you what happens with the result. So this was one that surprised us a little bit. We took the original kind of setup with the fibres aligned along the x-axis and turned down the kind of cell affinity so that the cell collagen interaction dominated more. And what we actually ended up with was aggregates again, but they had kind of swapped places from where they originally were. So you can see in the simulations what seems to happen is that the cells pull on the collagen, which means that the little clump that starts off on the top right will start to move left. And as the cells move to the left, they're pulling on the collagen and kind of pushing it behind them. So you end up with sort of a hole in the collagen, and the cells kind of clumped in that hole, which is kind of very unusual and we were a bit surprised by, but no matter how many times we tried to do it and destroy this, it kept happening, so it seems like that is actually what happens. And then, well, we did, I suppose, the obvious things. We started to look at turning on some of these parameters that we previously turned off. So we have our control simulation again with the fibres originally along the x-axis, but now we're going to turn on this tension in the fibre direction, so we turn that up to 10. And so when that happens, you can see that your original clumps start to elongate along the x-axis, kind of in the fibre direction, and you get much more sort of realignment of the fibres. And you can play with this parameter mu1, this fibre tension, and so we did that and looked at what we called the anisotropy ratio, which is essentially the sort of the ratio of the long to the short axis, or the sort of parallel to the x-axis and parallel to the y-axis axes of the clumps. And so, sorry, the reciprocal of what I just said. And so you can see that, well, as you ramp this thing up, you tend to get the aggregates elongating more and more along the x-axis. And so that was the effect of mu1. A similar effect when we turned up mu3. You get, again, this kind of elongation in the fibre direction, but less reorientation of the fibres in this case, apparently. Then, yeah, as I say, we were just playing, so we did things like, well, we turned our fibres around to 45 degrees again, and we turned on all of these anisotropic material properties, and we turned on the anisotropic drag, and then you can get your two clumps to elongate into a nice kind of long string across the middle of the square. And you can do things like have a initially varying fibre direction. So here's our initial conditions. So now instead of having just one kind of direction all the way through our domain, we've got it sort of spatially varying. And when we took our control parameters, but then turned on mu1 a little bit and turned on the anisotropic drag, we were able to get this kind of thing where you've got clumps, but their axes had kind of tilted. And then we took that initial condition again and ramped up mu1 even more, and we got things like this. And this is extremely puzzling to me, because you don't seem to get any. You get these kind of vertical stripes, but you don't seem to get any realignment of the fibre vertically. Although it's fair to say that there is realignment from here, right? It starts with a kind of with this variation, and ends up nearly parallel to the x-axis again. But it's not what I expect in terms of the fact that your cell distribution looks like a vertical stripe. So again, we tried running this and sort of little changes in parameters around this to see if something was going wrong with our simulation, and I don't rule out that it isn't, but as far as we could see, this is the result. So really, I think I want to conclude now. what we did was kind of just, we took a sort of a speculation, which was lots of people have done things looking at how chemical signals can give you different sorts of patterning in tissues, sort of whether it's very classical things like Keller-Segal or more advanced things. There's lots of kinds of different models out there, but we were kind of interested in, well, can you get pattern formation just produced mechanically? And we had this model that we came up with just sort of play with this idea and see what it could do. So we wanted to include the effect of fibres on the cells and how those fibres might affect the properties, the mechanical properties of the ECM itself. So we were able to, so then we made this assumption about how cells pull on the extracellular matrix, and we're able to do a little bit of approximation here to make things a little bit easier, assuming that each cell only has some small range of influence. And we were also able to do a little bit of analysis that suggests these kind of anisotropic diffusion equations that people have done before are kind of recoverable from what we did. So numerical results do seem to indicate that that these things are important. Fibre alignment, anisotropic ECM mechanics, these are important, and they can give you interesting different patterning in your cells. Obviously we need to investigate this with a much more sort of systematic kind of way, rather than the kind of scattergun sort of, oh, that looks interesting approach that we've got at the moment, and that's something that we want to work on. There's also things that we've ignored in here which might be interesting, so the role of cell proliferation, interactions between these kind of mechanical signals and other chemical signals that might be involved, interactions between different kinds of other cells that you might have if you're trying to grow a particular kind of tissue. So you might not just have a single cell population there, you might have others. So one of the things that Anna and Carlos were interested in was in adding fibroblasts to their mammary cells, because they seem to produce the ducts more sort of reliably. So again, there's all sorts of interesting kinds of ways that you could take this, and this has obviously been just something that's very preliminary. So I think that's where I'd like to leave it for now, and yes, thank you very much all for your attention.
In the collagen gel in vitro experiments that you mentioned at the beginning, is it possible to measure any of the parameters that go into your model?
No.
Not
well,
possible?
Well, I think
possible,
yes, possible, but no one actually has done so. So one of the things that Rosemary and I are interested in is looking at, some simple classical things like what happens if you take a layer of this stuff and squeeze it, what happens if you take a layer of this stuff and pull it, what happens if you try and push it through a channel, what happens if you try and put it in a cuvette kind of device. Look at these kind of classical things, and can you extract some of these, the mu1, mu2, mu3 from doing that? Yes. So I think
it's rather good to collaborate with the relevant experimenters to do that.
Yes. So Rosemary has some collaborators in Warwick who are not actually interested in collagen gel, but are interested in other fibrous materials, and they do have devices where they do these things experimentally. So we're trying to get at some of these things, but it's a question of the people who grow the tissues aren't always interested in doing sort of rheology of the gel and so on and so forth.
You need a few beers with them first, but yeah.
Indeed. Yeah.
If I understood correctly, the length scale that you're working on is many, many cell lengths, correct?
Yeah.
And yet you put in a sort of global orientation of the ECM. What happens if you would start with it essentially random over a larger scale?
Well, if it's essentially random, this
showing us that on a short scale, yes, they are aligned, but on a long scale, it looks isotropic.
0 to 1 rather than 1 everywhere.
Yes, so I think that's a very valid and important criticism, because there is the sort of degree of alignment kind of problem at each point. We're kind of assuming at each point in your gel you can say there's a sort of a perfect alignment, and I know what my alignment is there. And as you correctly point out, that's not really the case, that there's some variation in alignment because you'll have fibres crossing each other, and so a probabilistic kind of distribution where you have some probability density that says, you know, at each point I have the probability of finding a fibre with, you know, angle whatever is a better way to go, I agree, but also adds complication.
I have a related question, because it seemed that the change of the alignment, the initial alignment, was relatively little, and so I was wondering what happens if you turn up the parameter that will that will reorient the. So, for example, if you increase the force that the cells apply on the matrix, such that the realignment is going to be stronger.
Yeah. So all of the, well, I think for most of the pictures we were showing there, so the effect of ramping up the kind of the cell collagen force is equivalent to ramping down the gamma in our dimensionless system. And so apart from that one simulation, we actually looked at pretty large gammas in most of those simulations. So again, that's something that is interesting and needs to be more kind of systematically looked at. Yeah. So I don't know what the effect is the answer, but we need to.
Out of curiosity, what numerical method was used?
So this is where you probably need John, but I think he was doing a finite element method. I don't know all of the details because I'm the world's stupidest numerical analyst. I can just about do some basic finite difference stuff, but that's about it.
No more questions. Thank you.
Thanks.